I just wanted to inform you that this event will be recorded and will also be available on Umpa YouTube channel. So if you have your friends and colleagues and you want to share the today's science cafe on innate immunity, please you are happy to share that YouTube channel with your friends and colleagues. And you can also follow us on UMass Med Postdoctoral Association on Facebook, Twitter, and Instagram at UMass Med Postdoc. And before I move on to introduce our speakers for today, I just wanted to mention that the views presented here today's presentations are solely of the presenters and not necessarily of UMass Medical School. And if you have any questions during the talk, please type them in the chat box, and we'll also send out a link for the survey at the end of the event. So if you have any suggestions for the upcoming Science Cafe, if you want to hear on certain topics, please do tell us if you wanted to hear on certain topics, and please fill out the survey at the end of the event. So moving on to our first speaker for today, that is Havisha Honwad. She's a final year graduate student in Xavier Irozaki lab at UMass Medical School. If you wanted to connect with Havisha, you can follow her Twitter and Instagram at Havisha Honwad. And and today she'll be talking about the macrophages and their role in immunity. So let's hear more from Havisha. Havisha, please take it away. Let me just stop sharing.
Thank you, Preeti. I'm just going to share my presentation. Right. Is everyone able to see this?
Yeah, that's look good.
All right. So hi, everyone. My name is Havisha Honwad. I am a graduate student in the Irozaki lab in the MAPS department. And I'm going to talk about what I've been working on for the past few years and what I've currently been working on. So in our lab, we study how immune cells or bodies respond to infections or infectious agents. And to study this, we use models like immune cells from experimental mice or little worms called C. elegans to study the whole body responses. And we have that here up here in our logo. So immunity or immune response in our body has two main arms, right? There's the innate immune response, which we're born with, and there's the adaptive immune response that we develop over time in our lives. So innate immune response is considered as the first line of defense against germs or infectious agents or foreign particles. It is a quick response, it acts broadly, whereas the adaptive response is against specific targets or antigens, which are present on infectious agents. So our body specially produces antibodies against specific targets or antigens, and so it is comparatively a slow response as compared to this innate immune response. But here, there's memory involved. So whenever there's an infection which has already occurred before, the body can identify it and fight it. But what is crucial for mounting an immune response is that the immune cells in our body need to perform their functions properly. So understanding how immune cells regulate their actions or functions is very informative to understand pathologies and also to devise treatments against infections. So for today, let's focus on innate immune responses and the cells that are involved in this interplay. And since we're talking about cells, I want to ask all of you, have you ever thought about the size of the cells in our body? And be it blood cells that are red blood cells that make up our blood, or other immune cells like B cells that produce antibodies, macrophages in the innate immune response, or T cells, which are actually killing invaders or pathogens. So can you guess out of these cells, which cells are the largest in size? We can have a quick poll to get all of you to interact maybe.
We'll give it 15 more seconds.
Okay. Interesting answers.
All right, let's
Right.
see the
I think
results.
everyone in the audience is very well versed with what I'm going to speak about. Can everyone see the results? Like 80% of the audience is right. So that's awesome. People know this already. All right. So macrophages, right? Macrophages are the largest cells in our body, and they're so big that they're about 21 microns in size as compared to red blood cells, which are super tiny, and B cells or T cells, which are 10 to 12 microns in size. So let's talk more about macrophages. So these are white blood cells. They play a crucial role in inflammation and infection. And the main role of these cells is to eat. And their name actually means big eaters. So what macrophages do is that they surround particles that they want to eat, engulf them, and suck them right up. This process is called phagocytosis. So macrophages are professional phagocytes. They're constantly surveilling our body. They're behind the actual physical barriers, the skin, the mucus, or the gut. And they're looking for foreign particles or invading bacteria or viruses. And this is what makes them important players in the immune system. Because as they're doing this job of cleaning up or eating debris, they decide when it is needed to initiate an immune response. And while doing that, they're able to secrete what are known as cytokines or messenger proteins to communicate with other immune cells in the body. And all of this helps orchestrate an immune response. And in fact, macrophages are also needed to curb or stop inflammation and repair tissues or heal wounds as they clean up the debris and overall maintain a balance or homeostasis in our body. So the function of a macrophage depends on surrounding signals and stimuli. And based on the environment, there are changes in gene expression that allow a macrophage to switch from a basal state to an activated state, which is referred to as macrophage polarization or activation. Studies over the years on macrophage activation have shown that there's a spectrum of phenotypes that these cells can change into, and all of these carry out specific functions. But to just simplify this concept, there are considered two major activation states. M1 state, which is a pro-inflammatory state where a macrophage is in fight mode, and M2 state, which is a pro-resolution or an anti-inflammatory state where the macrophage is cleaning up after a fight. So expression of the right genes at the right time in macrophages is very crucial. Sorry, I'm just going to hide this panel for you guys to see the titles. So expression of the right genes at the right time in a macrophage dictates the switch between these activation states and to perform necessary functions. So how regulation of gene expression takes place in a macrophage is what I have been really interested in studying. So brief background on the central dogma of molecular biology. This is a cell, and inside it is the cytoplasm that surrounds the brain of the cell, which is called the nucleus. And in the nucleus is present DNA, which is all of our genetic material. Now, gene transcription is the process which converts DNA into RNA, so converting genetic information to transcribable genes, and that is translated into proteins, which are the important molecules in our body. So in complex organisms like ours, like humans and mammals, there are specialized proteins called transcription factors, normally in the cytoplasm, but they travel into the nucleus, sit on the DNA, and transcribe a bunch of genes, a subset of genes, and that is dependent on when it is needed and how much gene expression is needed. So transcription factors do that. For example, when a macrophage senses pathogens around it invading infectious agents, a specific transcription factor called NF-kappa B translocates from the cytoplasm to the nucleus, sits on the DNA target site, and induces the expression of immune response genes to fight and kill the pathogen. Like these genes, which help the macrophage switch to an M1 pro-inflammatory activation state, or induce the expression of the genes needed to make cytokine proteins that are secreted out of the cell. So depending on the environmental signal or the kind of input, regulation of gene expression drives macrophage activation states, which have important functional consequences for health and disease. because a dysfunction in that might lead to immunodeficiencies, where the immune response is not sufficient to get rid of an infection, hyperinflammation, where it's the opposite, that there's too much immune response, or even autoimmunity, where the immune cells are attacking own body cells, which it's not supposed to do, and that causes damage. So a lot of research has been done on transcription factors like NF-kappa B in the context of infection. But what is new or what we have been studying in the lab is this other transcription factor called transcription factor EB. So we found that in mouse macrophages, when we infect them with staph infection, Staph aureus, this is a bacteria that causes skin infections, which I'm sure everyone is aware of at this point, but we can talk about it more later. Or just treating with molecules from staph bacterial cell walls, which is peptidoglycan, we can see TFEB nuclear translocation or TFEB activation that we say. This is a nuclear translocation assay that I've shown you. And in green is TFEB fused to green fluorescent protein. And that's a recombinant protein that we have in macrophages, which can help us visualize the translocation of this transcription factor and look at TFEB nuclear translocation or activation. And so this is the quantification in the form of nucleus to cytoplasm ratio. So we're looking at how much TFEB is in the nucleus as compared to the amount of TFEB in the cytoplasm. So we see TFEB activation, a new transcription factor. So why is this cool? Now, before this, TFEB was known to be activated during conditions of cell stress like nutrient deprivation or starvation and non-bacterial infection. So during stress, TFEB is activated. It translocates to the nucleus to drive transcription of lysosome and autophagy genes. So lysosomes are acidic molecules or acidic membrane-bound compartments found in the cells that contain proteases or hydrolases to break down nutrients or even other things that a macrophage ingests. And the process of autophagy is this orderly degradation and recycling of nutrients within a cell. So especially in macrophages, this is very useful because it helps degrade all of the phagocytose stuff, which they constantly keep eating. TFEB is also known as a master regulator of genes that are responsible for cell clearance mechanism because it helps in nutrient recycling and also formation of new organelles within a cell. And so I want to highlight the fact that TFEB is at the center of cell metabolism or cell signaling. It is an important sensor able to sense changes or different inputs. And this is why it is important to study TFEB-mediated regulation of macrophage function. Additionally, TFEB is expressed in different tissues in your body. So influence of TFEB can touch a variety of disease and pathological states. For example, in the brain, it helps prevent accumulation of protein aggregates, which causes Alzheimer's. And overall, it helps people age better because it clears toxin, waste, and recycles nutrients, right? So based on TFEB's known function, we observed that both staph infection or peptidoglycan treatment in macrophages showed associated increase in lysotracker, which tracks lysosomes, as well as lysosomal protein, LAMP1, showing that both staph or peptidoglycan-induced TFEB activation was biologically significant. So with this preliminary data, we set out to address two main questions, right? Question one, what happens to a macrophage response against Staphylococcus infection in the absence of TFEB? And second, what is the intracellular pathway or signaling involved linking bacterial infection to TFEB activation? So we performed a technique called RNA sequencing that helps measure overall gene expression in macrophages. And we saw differences in macrophages that don't have TFEB as compared to wild type or normal macrophages. And consistently, there was a decrease in the amount of cytokines, right, the protein messengers that are secreted in TFEB deletion macrophages, all of these three, compared to wild type macrophages in response to the staph infection. And this we measure with a technique called ELISA. Most importantly, the absence of TFEB reduced the bacterial killing ability of macrophages compared to normal macrophages. And this is using a technique that involves counting the number of bacteria that a macrophage engulfs or has eaten and which are internal. So I've included this image to show you the cool picture. This is just light microscopy, basic microscopy about 40x magnification. These are all macrophages. And the white dots that you see inside the macrophage are the staph, the bacteria. So there's also more data that I'm not showing here, but we did a series of experiment and came up with a model. So we propose this model of what we think is happening after staph infection. And what we saw is that TFEB activation, right? TFEB going into the nucleus is happening because of oxidative stress that eventually leads to a calcium ion release and regulates TFEB activation in macrophage and associated defense function. So studying macrophage TFEB to show how it is emerging to be an important integrator of many different signals or inputs has been the main objective of my thesis. So the other input that I'm currently studying is neurotransmitter acetylcholine or cholinergic stimulation. So changing gears completely, this is an interesting different input that I'm currently focused on. So since I mentioned that we also use C. elegans or little worms in the lab to study defense response against infection, apart from mouse macrophages, there was a recently published study in this C. elegans model system showing that in C. elegans, whenever there's staph aureus infection, there seems to be a release of acetylcholine neurotransmitter that regulates antimicrobial response or defense response against the infection. Now, worms don't have special immune cells like macrophages, or they don't have special transcription factors like the NF-kappa B, but they have TFEB in their intestinal cells. And so we asked the question, does acetylcholine or cholinergic input have any effect on mouse or human macrophage TFEB? And in fact, there's been a lot of studies on the neuroimmune link, interactions between the nervous system and the immune system. So we know that mammalian immune cells express neurotransmitter receptors. So neurotransmitter acetylcholine functions via two cholinergic receptors. One is the muscarinic or G protein coupled receptor, or the other is nicotinic or ligand gated ion channel receptors. So the ligand here is acetylcholine, the neurotransmitter, and stimulating this receptor causes an influx of calcium ions. Now, when I talked about the molecular link between bacteria and TFEB activation in macrophages, I did mention a calcium ion release, right? So because of this connection, we asked, is macrophage TFEB activated if we treat them with acetylcholine or acetylcholine-like drugs? And so in fact, treating macrophages with acetylcholine analogs like aricoline, which activate all kinds of acetylcholine receptors, be it the muscarinic or the nicotinic receptors, we saw that it causes TFEB activation. So here is the same system that I showed you before, TFEB fused to the green fluorescent protein. And here in the control condition, we see nuclei which are empty little holes, right? So that's showing us that TFEB is in the cytoplasm and not the nucleus. But here in aricoline treated cells, TFEB translocates to the nucleus and so is activated. Additionally, we used this acetylcholine specific, like one receptor, nicotinic receptor called alpha-7. And there is a specific drug that stimulates this receptor called PNU. So just like aricoline, PNU also showed TFEB activation. We have a positive control here, which is the starvation condition, since we already know that starvation causes stress to the cells and sends TFEB to the nucleus. So this is a good positive control to have. And what is interesting is that one nicotinic receptor, this alpha-7 nicotinic receptor, if we inhibit this with a specific drug called MG624, it just totally abrogates this TFEB activation effect that we see with aricoline or even the specific receptor activator. And what is interesting is that TFEB activation is associated with also increase in lysosomal acidity, right? Because TFEB is controlling lysosomal genes. So we see PNU or acetylcholine analogs increasing lysosomes in macrophages and inhibiting that one particular receptor of one particular acetylcholine nicotinic receptor also inhibits this lysosomal increase that we see. So with this preliminary data, I'm currently working on this project and asking a bunch of related questions as to what this cholinergic stimulation is doing to TFEB and associated macrophage phenotype or function. And with this, I quickly want to summarize the two main points that I talked to you about. One is that TFEB regulates macrophage response to staph infection, because absence of TFEB affects macrophage gene expression, cytokine secretion, as well as the killing ability. And secondly, there's this new input that seems to cause TFEB activation in macrophages and causes associated lysosomal increase, which currently I'm trying to dissect. And so with this, I'd like to thank my lab, my PI, Javier, the department, my thesis committee, and especially UMPA for providing me with such a nice opportunity to present. And also a big thank you to the audience and everyone who's attending and participating. So I'm happy to take questions if there are any, and thank you for listening.
Thanks, Havisha. It was really interesting talk. So we are having a couple of questions. Yedwan from our audience is asking, does TFEB deletion affect the normal function of TFEB other than macrophages?
I'm sorry, I'm just going to quickly ask you to repeat that question. Sorry, I was just trying to.
So the question is, does the TFEB deletion affect the normal function of TFEB other than macrophages?
Normal function of, sorry, what?
The question is, does the TFEB deletion affect the normal function of TFEB other than the macrophages? That is the question.
Sorry. Okay. So TFEB deletion in mice is actually embryonically lethal. So it seems that it performs important functions, which is why it's interesting, right? So we do deletions in specific compartments or specific tissues. For example, we deleted TFEB only in the macrophage line or only in the macrophages. So to avoid any other effects. So since we know that TFEB is important for the whole organism, we're trying to study more about it. So yes, there are effects, but which is why we're just focused on this one system.
Okay, so the other question is from Sumitra. She's asking that GFP is known to have high half-life. Based on it, do you think if it is possible to know whether the TFEB was expressed transiently or not?
Yeah, so that's true. What we do is this is a cell line. So we have TFEB overexpressed, right? So it's always crucial to look at endogenous expression of TFEB, and we do that. So I just didn't include the data here because we didn't have enough time, but we have similar results with endogenous TFEB activation.
Okay, so one more question is there. How does starving the cells mentioned was positive control as induces acetylcholine to translocate to the nucleus? It was asked by Disha Khanna.
Okay, I'm sorry, that was, I think that there's a confusion there. So the starvation condition was used as a positive control. So that's separate to this acetylcholine treatment that we did. The whole point of using starvation was just to show that there is TFEB nuclear translocation, just like a positive control in the acetylcholine treated macrophages. So starvation and acetylcholine are currently not connected, at least in my study.
Okay.
Yeah.
So Kay is asking, are there other type of macrophages, and does this phenomenon stay true to all of the type of macrophages?
There are many types of macrophages. Currently, I'm using one system, which is the bone marrow derived macrophages, and immortalizing these bone marrow derived macrophages for creating this GFP fused TFEB cell line. We do want to explore tissue specific macrophages and other like macrophages which have specific tissue functions. But I think the first step is to establish this link or phenomenon in one system and then go on to the others. So hopefully this is true to most macrophages. TFEB function is definitely true to most macrophages, is what studies say in the literature.
Okay, and then Alicia's question is TFEB induced by all type of bacteria and macrophages, or only one type or a certain type of bacteria?
Yeah, so we did try gram positives, which is staph is an example. Staph aureus is 1 gram positive bacteria. We've also tried to study salmonella, which is a gram negative bacteria, and that also induces TFEB. Plus, apart from us, there are more research groups which are studying this TFEB activation induced by infection. And it seems like a lot more bacteria induced TFEB activation. So it's not only restricted to one certain type. So it's interesting to study.
Okay, so we are having 2 more questions from Imancula. We are having adipose tissue macrophage polarization has been shown to affect glucose tolerance.
Mhm.
There have been implications that catecholamines might affect the immunometabolism and play a role in diabetes. Do you know if there are any implications in literature of association of TFAB and diabetes?
For sure, there's a lot of connections with TFEB and metabolism, just because it regulates so many cell clearance mechanisms. So there's more knowledge than I am aware of at this moment, because TFEB has just suddenly popped in the literature over the last decade or so with functions like that. And so it's going to be interesting to really study this. And now TFEB is also used as sort of like a therapeutic. It has a lot of therapeutic potential. In fact, they're developing strategies to activate TFEB or even inhibit TFEB, because if you increase TFEB expression, that is also bad because it might cause cancer, right? Because it's causing the cell to recycle nutrients and constantly have new things ready. So it's a fine balance again, which is why I'm at the center of looking at balance, either with macrophages where it's immune response, or TFEB in the cells. So yeah, it's in fact coming up.
Okay, that's all of the questions. And thank you, Havisha, and it was a great talk. Thank you.
Thank you so much
Yeah.
for listening.
So much.
I'm really sorry for the first question. I think I didn't listen to it, but now that I read it, TFEB is expressed continuously. It's not a transient expression. Sorry about that. That's all.
Yeah. Thank you so much, Havisha. So if any of you still have questions, you can get connected to Havisha on Instagram or Twitter and ask your questions there. And we'll move on to our next speaker now. Okay, so our next speaker for today is Dr. Theresa Ramalho, and she's a postdoc in Cole Hens lab. She did her PhD from University of Sao Paulo in Brazil, Southeast Brazil. And if you want to connect to Theresa, you can follow her Twitter and the Instagram at Ramalho Theresa. Again, I wanted to mention that this event will also be recorded and would be available on our YouTube channel, UMPA YouTube channel. So if you want to share it with your friends and colleagues, please share the link with your friends and colleagues. And I'll move on to Theresa, and we'll hear more from Theresa now. Let me stop sharing.
All right, can you see my slides?
Yes, we can.
All right, so thank you, Sneha, for the introduction. Yeah, so today I will talk about this project that was conducted in the laboratory of Professor Ricardo Gazinelli in the Department of Medicine. And I spent there 2 years in this lab, and in my project, we try to understand the metabolism of the host infected with Plasmodium that triggers malaria. And we try to understand how the metabolism of the host is able to interfere with the immunology of this host, and how that increases the severity of the disease. Let me get the pointer. All right, so to start, I want to introduce you guys to the malaria disease. So the malaria is a complex disease because it has more than one component, as I will show you. So the disease is caused by the infection with this parasite that is called Plasmodium. And the parasite, the Plasmodium, is transmitted by a vector that it's called Anopheles, which is a mosquito. And Anopheles is able to infect hosts with this parasite, and the host can be birds, reptiles, and humans. So when this host is infected, this is the cycle of the disease that happens. So here, Anopheles is biting the skin of the host, and the parasites are inoculated in the stage of sporozoites. And these parasites, they go to the liver, and from the liver, they reach the bloodstream. In the bloodstream, these parasites, they infect the red blood cells. And these red blood cells, they can reach the spleen, where we'll have the immune response against the Plasmodium. Or these red blood cells, they can have a lot of proliferation of these parasites, which ends up exploding these red blood cells. And when these parasites go back to the bloodstream, they become available to a new bite from Anopheles, which will continue the cycle of the disease. So because the immune response against Plasmodium sometimes is affected, sometimes is not very effective, we have a huge incidence of malaria in the world. And I want to make sure that you guys are aware, a little bit aware of the disease. So I want to ask you guys if you know which region of the world most accumulates the incidence of malaria. So this is the poll that you guys can choose your answers.
We give it 15 more seconds.
Right. And it's over. I think most of the people are correct. How can I stop? Okay, so most of the people are aware of what's going on in the incidence of malaria. And yeah, so Africa is the continent that most accumulates the incidence of cases. And after Africa, we have Asian and Eastern Mediterranean region with 10 and 5% of incidence of the cases, and Western Pacific region, Americans and Europe contributes with less than 2% to the malaria cases in the world. Regarding the deaths, again, Africa accumulates most of the deaths, and it's important to mention that these deaths, they happen mostly in children under 5 years old, exactly because the immune response of these children is not mature enough. Yeah, in the other regions, they contribute with less than 3% to this, to the incidence of deaths in the world. There are strategies that try to reduce the burden of malaria. So there is this vaccine called Mosquirix, manufactured by GSK, that started to be given to children in Africa since last year. There is this other vaccine being developed by Oxford in phase 3. And BioNTech promised that by the end of this year, they're going to start their clinical trials with this vaccine of RNA. There are also medications that help to prevent the infections in the host, and also medications that treat the host infected. And these medications, they target the parasite and not the host. And utilizing these two strategies, World Health Organization has developed this plan that will try to reduce the incidence of malaria at least in 90% until 2030. And while these strategies are still not as effective as they should be, remains for the research groups to try to understand the mechanism, the biological mechanism of the diseases as malaria, to try to reach new additional treatments to improve these conventional therapies. So so in the research, we, as Havisha mentioned, we try to understand immunology in this perspective, in innate and adaptive immunity. But today I will focus a little more in the dendritic cells and in the T cells, which are the cells that we studied in the metabolism of malaria. So the group of Professor Ricardo, they understood very well how this feedback happens between the glucose levels in the blood and also the parasite proliferation. So they basically found that there is a positive correlation going on here. And in this project, we have found that the spleen is this organ that uptakes a lot of glucose from the bloodstream. And because of this glucose uptake that happens in the spleen, we have these low glucose levels in the blood. And I just want to mention here that the spleen is exactly the organ where we have the immune response against Plasmodium. And this immune response is composed by innate immune cells and also adaptive immune cells. This is the mechanism by which the immune response happens against Plasmodium in the spleen. So here we have the spleen of these mice full of parasites and inflammation going on. So in this context, we have these dendritic cells, which are part of the innate immune response. So these cells, they live in the liver and they uptake the infected red blood cells. So these cells, they are able to process these antigens, just like Havisha showed in her presentation. And from this processing, from phagocytosis, these cells are able to literally introduce these antigens to lymphocytes, which are part of the adaptive immune response. When these lymphocytes become aware of these antigens that were introduced by the dendritic cells, they get activated and they start to release these pro-inflammatory factors that start to recruit new dendritic cells to the spleen. So when these new dendritic cells reach the spleen, they become activated, they become mature, and they migrate to the brain, where they're going to conduct a lot of inflammation and will end up killing the mice. So knowing that the spleen is this organ where we have the immune response going on and a lot of, and also this organ consumes a lot of glucose, we want to understand how this consumption in the spleen affects the immune response in malaria. So before we go to answer this question, I want to make sure that we are all on the same page, and I want to talk a little bit about metabolism. So the concept of metabolism is exactly this reaction that happens through an enzyme that accelerates the processing of a substrate into a product. So the product of metabolism is called metabolite. And to exemplify this, we have the spermatozoa, which is a eukaryotic mammal cells, which is composed by mitochondria. It has a lot of mitochondria in the cells. And basically this organelle it has a lot of enzymes which are going to convert glucose into ATP. So basically for mammal cells, glucose is the substrate and ATP is the product. And ATP is the basic unit of energy of our body. And that's how we have energy to survive. In the case of immune cells in the spleen, this is the graph of an spleen. And here we have an immune cell, a dendritic cell, which is uptaking glucose. But in the case of immune cells, when they become activated, their mitochondria starts to act weird. So instead of promoting energy, instead of releasing ATP as a product, this mitochondria starts to release acids and also other craps that will end up activating innate immune sensors, just like the ones that Havisha explained. And so to answer the question that we're trying to understand how this glucose uptake in the spleen affects the immune response in the spleen, we decided to start evaluating the metabolites that these dendritic cells are synthesizing. So here in this graph, we have the fold change, the amount of increase that these metabolites are accumulated in the dendritic cells compared to the cells of non-infected mice. And here we see that most of the metabolites that are accumulated in the cells are metabolites that are released from the mitochondria, from a sick mitochondria. And this itaconate is the metabolite that is the most accumulated in the cells. And basically, we in the validations of the tests, we observed that itaconate indeed is synthesized through the metabolism of glucose in the mitochondria. And this itaconate, according to the literature, is a metabolite that is synthesized by innate immune cells, and it has bactericide and anti-inflammatory properties. And because of that, itaconate is being a good candidate to treat tuberculosis and also sepsis by utilizing it in an exogenous treatment. Yeah, so now that we know that these dendritic cells are able to uptake glucose and synthesize itaconate, we found out that this itaconate can be released by the cells and it can reach the bloodstream. As we can see here in this graph, after a week that the mice are infected, we can detect the itaconate levels increase here in the blood of the mice. And this amount of itaconate is consistent with the amount of parasites. So the more parasites the mice have, more itaconate they have. And the same is true for humans, for patients infected with Plasmodium from the north of Brazil. Here, each dot of this is a human, and we see that when they have malaria, they have more itaconate than non-infected humans. So to try to understand what exactly itaconate does with the immune response, we utilize these mice that are not able to synthesize itaconate. They basically don't have the enzyme that makes itaconate. And we compare itaconate-free mice with mice that produce itaconate in both conditions, non-infected mice and Plasmodium infected. And here we see that when mice are not able to synthesize itaconate, their lymphocytes, they become more activated. And because of that, these mice that do not synthesize itaconate, they have a better control of the infection. They have less parasites. And so here to represent these results, we have that in the spleen of infected mice, where we have a lot of proliferation of Plasmodium, the dendritic cells uptake a lot of glucose, which ends up releasing itaconate. And when this itaconate is released to the bloodstream, they limit the activation of lymphocytes, which ends up improving the capacity of Plasmodium to proliferate. So from this, I have some take-home messages that it's basically that the glucose metabolism in the immune response in malaria is very important to propagate the severity of the disease. And from that, we suggest that a ketogenic diet based on protein and fat can be a better approach compared to a diet that is composed by carbohydrates. And we also suggest itaconate as a biomarker for the severity of the disease. And also, if we could target itaconate to reduce its levels in the bloodstream, maybe we could help with the efficacy of other conventional therapies. So I want to take advantage here of the space and call attention for this humanitarian emergency that is going on in Brazil, in these Yanomami tribes in the north of Amazonia. Basically, there is a lot of illegal mining going on in these regions, and these illegal practices are rising every year by year, and there are a lot of consequences behind that. And an important consequence of that is malaria, because these people, they are basically isolated from the world in their own culture, since the world is a world. But now with these invasions of rivers, invasion of the forests, that contributes to the contamination of the rivers, there is malnutrition. So all that contributes to a worse incidence of malaria. And these people are literally dying of malaria in a country that is the biggest producer of chloroquine. So it's important for today, I think, for everybody to become aware of the situation. And that's it. I would like to thank you all for the attention. Also want to thank all the people that got involved in this story. Also thank you, UMPA, for organizing this event. And also thank you, I want to thank the agencies that helped me to develop this project with money and the space. And I'm ready to take the questions.
Thanks so much, Theresa. It's a wonderful talk. And on chat, please type in your questions. And okay, here is one question. So it's from Raphael. Hi, Theresa. I worked with Ricardo more than 10 years to increase work. I currently work on gut microbiota impact on anti-malarial immunity. Do you know if the knockout mice have a more protective microbiota, or do you think the mechanism is more macrophage based?
That's an excellent question. We don't know the microbiota of this mouse. I don't think anyone ever studied the microbiota of this lineage. It's available on JAX if you want to to collaborate with some experiments, we can find out together. But I would say that in terms of microbiota, maybe if we in the knockout could be a little more I think that it could be a protective microbiota, considering that you have less inflammation, you have less parasites to take care of. So I think it would be healthier by consequence. I don't know exactly if the itaconate itself could directly modify the microbiota, but I think as a consequence of the system, the microbiota could be healthier in the knockouts.
Okay, I got a question from Alessia. So how effective are the currently approved vaccines and treatments? And do they have side effects? And also, why is the chloroquine not available to everyone? Is that lack of awareness, cost, or accessibility?
Great points, Alicia. Great points. So the vaccine that it's going on right now has an effectiveness of 50% for infections. Mortality is still being evaluated. And what else? Regarding the treatments, effective. So the treatments, they are effective to prevent the infection, especially, but there is a lot of resistance going on. Like the parasites are starting to get resistant, and especially if the host is not healthy, this resistance simply doesn't happen because the immune system is not strong enough to fight that. And if you don't have a good immune response, doesn't matter if you are targeting the parasite, because the parasite is still going to be there, even if it's weak. But about the side effects, yeah, so for the vaccines, they guarantee that there are no big side effects, like relevant to stop the trials. And about the medications, those medications, they're very toxic. They, like, if you take it's easy to vomit, and it's a very high dosage, and again, there is a lot of resistance. And in the case of chloroquine, that it's not available. Actually, chloroquine it's a complex story of the chloroquine, because during COVID outbreak, people bet a lot on chloroquine, that chloroquine could treat COVID. And in my country, we had a crazy president that for years suggested and told people to really take chloroquine to treat COVID, which is crazy. I agree, but because of that, the basic needs for chloroquine started to be out of the medications like lupus and malaria. So yeah, so chloroquine is available, but during COVID we had these complications because of where to focus the treatment, and in the case of cost accessibility, this is also an issue because the Amazonia is a closed environment that it's hard to reach. But these areas, they used to have a very good public system that would provide everything these populations need. But because of this crazy president, we had all this basic rights were removed from these people. But now we are returning to normality and civility. So maybe we'll take more care of this people now.
Okay, we got another question from Raphael. Are you looking at other metabolites after itaconate?
Yes, I checked succinate through the action of its receptor in lymphocytes, but I didn't find any good correlation between the effects of succinate and itaconate. So I don't think itaconate is acting through succinate. But right now I am moving to a new project that I am investigating, not exactly itaconate, but the effects of the metabolism downstream itaconate. I'm basically interested in the post-translational modifications that happens downstream itaconate.
Okay, I think that's all the questions. Thank you so much, Theresa.
Thank you.
That's great. Thank you, Havisha, Theresa. This has been a great evening. Thank you so much for discussing your great research with us. If anyone has any more questions, please feel free to reach out to us or reach out to Theresa and Havisha specifically. We would really encourage you to join us for all our science cafes. We do post these cafes on YouTube, so you can find the link for our YouTube channel and subscribe to it so you know about all the talks that we've had and the ones that are coming up. And we also will be sending you a short survey, so please do fill it so we know your thoughts about the event as well. Thank you so much. Thank you.